afternoon, everybody. My name is Alessia Tatti, and today I'm going to show you a chapter of my master thesis regarding a tool I developed for the study of fungal biodiversity. Okay, so why I decided to study fungi? Because they are usually neglected organisms, even if they have a crucial role in biological ecosystems. So they could be decomposers regulating nutrient and carbon cycles. They could be mycorrhizal symbionts connecting the activity of separate plants and enhancing the primary production. They could be also parasites regulating the population dynamics of the hosts. And when we think about fungi, we usually think about mushroom, but there are a lot of other microfungi, and we know actually about 20 phyla of them. But even if they are so important in ecosystems, we know only 1% of their biodiversity. And so I thought, how can I contribute to their knowledge? And so I thought of the primary database online that have a lot of sequences from amplicon sequencing now. And since there is a lack of a comprehensive tool for the study of barcode, and with a willingness of learning how to code in Python, I decided to develop a new Python tool subdivided in 2 classes and 42 methods. So in the first part, the tool is going to provide you automatically a data set and a data frame with all the metadata associated to those sequences. And the data set is given you already formatted in a FASTA file. And it could be done both with a keyword search and a blast search. In the second part, the code is going to align the data set and then automatically clean the alignment. And then each single gene alignment could be concatenated, different markers, I mean, and then the code is going to give you a maximum likelihood tree. And then it can do also a barcoding gap analysis, doing first a pairwise distance matrix and then giving you a barcoding gap histogram and a box plot of your data. So this is an example in the upper part of a call of the build dataset method, while in the lower parts of the screen you can see before and after the automatically cleaning of the data of the alignment. So how we can do it: removing 2 long sequences, 2 short sequences, poor quality sequences, and then trimming head and tail of the alignment, removing sequences with too many gaps, then removing also the odd sequences, meaning those that opens new gaps, and then removing the only gaps column. Then the code can concatenate different markers. And I thought of a new way to concatenate them, because the existing tool permits you to concatenate on the basis of the name of the sequence. But in this way, you usually can create chimeras. So to avoid create chimeras, I thought to use the metadata of the sequences with the identifiers, meaning some codes, specific code for a sample. So in this way, you can not create at all chimeras. And also you can try to create the best chimeras if you want to concatenate more sequences, because not always you have the identifiers. And to create the best possible chimeras, the code is going to use the sampling place and the host. And then in the final part, the code is going to do the barcoding gap analysis. So the barcoding gap is the gap existing between the interspecific distances distribution and interspecific distances distribution. And then you can plot in the histogram the species you are interested in. And here, for example, you can see two different species in blue that actually belong to the intraspecific distribution. So they are not different species. While two morphologically very similar species in red that actually belong to the interspecific distribution. Then the code is going to provide you also maximum likelihood tree, so you can confirm those information. However, the barcoding app usually does not exist. The two distribution overlaps, and this could be because of paraphily, wrong assignment, taxon sampling, bad alignment, and so on. So I thought, what are those outliers of the two distributions? And these are actually the more interesting stuff I found. So they are in blue, you can see Cantarellus pallens versus Cantarellus cibarius. They are two different species. However, some sequences belong to the intraspecific distribution. That means that there are some wrong assigned sequences. While in red, you can see some Cantarellus cibarius versus other Cantarellus cibarius in red, and they are actually probably cryptic species. How I know this? Because the code is going to do some blast search automatically, and it gives you the higher ident that he found. And then in the upper part of the interspecific distribution, usually you can find some contamination of microfungi. And this is an example of what a small taxon sampling can do. So if you have a small taxon sampling, the code can't recognize the outliers. And so you have this overlapping distribution, while with a higher taxon sampling, the code is going to recognize them and remove them from the analysis. So you have a clean histogram. You can also apply the code in whatever organism you want. And we tried it on Aedes. And in this case, we were able to find out 3 cryptic species in Aedes japonicus and 1 in Aedes corecus. So to conclude, I developed a comprehensive Python tool for the barcode analysis that overcomes some time-spending procedures, such as the creation of a data set and the cleaning of alignment. And it gives you a new method for marker concatenation, and also it permits you to identify misled and cryptic species. However, it has some limitation, time-spending procedures such as the blast search. And since there is a lack of homogeneity in metadata, you can lose some information, and the code does a lot of assumptions. So the sequences need to be of good quality, and the identification of most of the species need to be correct. And then the code is not yet user-friendly, but we are working on it. And I want to thank my supervisors and also my colleagues at University of Padova, of Trento, and of course the Systematics Association. Thank you.